Dear students, I welcome you all. Today I am going to talk about pollen-pistil interaction. As you all know, in flowering plants, one of the most important steps in a reproductive process is the pollination. Pollination in angiosperms is said to be indirect pollination, since the pollen lands on the stigma much away from the ovule, which is well hidden and deep seated inside the ovary. Thus, in angiosperms, the pollen brings out a tube which runs through stigma, style and then ovary, reaching to the inside of the ovule, rupturing there and releasing the gametes for bringing about fertilization successfully inside the embryo sac. This is called as siphonogamy, as a pollen tube carries the gametes to the embryo sac for bringing out fertilization. Mere random landing of a pollen on the stigmatic surface does not mean that it will follow the sequence of the said events and bring out fertilization of the egg inside the embryo sac. Instead, there are many vital cellular and molecular interactions which occur between the pollen and pistil, which prove deterministic in allowing or inhibiting the pollen from bringing about fertilization. The first interaction between the pollen and the pistil is pollen adhesion. The stigma of a pistil is the site for receiving the pollen grains. The stigmatic surface shows a wide variation in morphology and exudation in different angiospermic plant species. The stigma in some species does not secrete exudates and is thus referred as dry stigma, while as in other species it secretes the exudates and is called as wet stigma. The wet stigmatic surfaces promote the adhesion of most of the pollen species, while as in dry stigmatic surfaces such as those of Brassicaceae, the pollen adhesion is a complex process which regulates the pollen acceptance. Thus, the control of pollen acceptance in dry stigmas is exercised at the level of adhesion. Let us talk about how this interaction is brought about in case of dry stigma. The adhesion of pollen grains with dry stigma is under polygenic control. In Brassica, pollen adhesion increases with time, recurring almost half an hour to achieve a maximum binding. It is a well known fact that in Brassicaceae, the pollen coat or tryphene contains components that are implicated in mediating a cell-cell interaction between pollen and stigma. The pollen coat finds an essential role in attaching the pollen with the stigma. Its absence has been shown to turn off the pollen adhesion. The pollen stigma interaction influences or changes the structure of both the stigma papillae and the pollen wall. The interaction causes such a change in the pollen coat that it results in the development of hydraulic continuity between the stigma papillae and the pollen grain. Studies of pollen adhesion carried out on Arabidopsis thaliana showed that the stigma of the species bind selectively the Arabidopsis thaliana pollen with a very high affinity, and this interaction occurs very fast, consuming only few seconds after pollination. The monocotyledonous pollen showed a very little or no binding capacity with the stigma of this species. Pollen from various dicot species showed a range of binding capacities, but this binding could be easily broken down or washed off by a detergent treatment. The binding of Brassica campestris pollen, which is a close relative of Arabidopsis, was no different than other dicots and was easily washed off by a detergent treatment. However, the binding of the Arabidopsis pollen and stigma was resistant to the detergent treatment, therefore indicating that pollen adhesion is a highly specific event. The genetic studies have shown that there are some mutants called less adherent pollen or LAP mutants, which show less binding with the stigma. The mutant called LAP1 shows a gross defect in the exene structure, which thus proves that it is the pollen coat which has a role in pollen adhesion. There are two stigmatic proteins which are indicated with a role in pollen adhesion in Brassica. Both of these are members of the self incompatibility S. locus gene family. These include S. locus glycoprotein SLG and S. locus related protein SLR, also called as SRP. The role of SLR1 in pollen adhesion was determined in transgenic plants by down-regulating its expression by antisense suppression. This resulted in the reduction of pollen adhesion, but the stigma retained the ability to support pollen tube growth. In wild type stigmas treated with anti-SLG and anti-SLR1 antibodies, the pollen adhesion was reduced drastically, therefore making it evident that these two proteins are involved in pollen adhesion. Further, these two proteins SLG and SLR1 are known to interact with pollen coat proteins in vitro. The pollen coat proteins are small cysteine rich proteins. Two members PCPA1 and PCPA2 are known to interact with SLG and SLR1 in vivo, thus confirming some role of these proteins in pollen adhesion. Now, let us come to the second interaction, that is pollen hydration. After pollen adhesion to the stigma, the pollen hydration is a critical event for the successful pollen germination or pollen tube growth. The process of hydration is supposed to have a divergent mechanisms in angiosperms, since there is a wide diversity in the nature of the stigmatic surfaces in the angiospermic group as a whole. However, the outcome of hydration seems to be common in all cases, which results in a wholesale reorganization of the vegetative cell. There is a substantial body of evidences which involve the long chain lipids as the agents which act as signals to stimulate pollen hydration. Mutants with defects in lipid biosynthesis in the stigma show defects in pollen hydration. The SER and POP1 mutants having defects in pollen hydration have defects in lipid biosynthesis. The exogenous application of long chain lipid trinoline, which is one among the exudate lipids, caused normal pollen hydration in POP1 mutants. The role of exudates in pollen hydration is best exemplified by Nicotiana, where the pollen grains which adhere at the top of the papillae above the level of exudates fail to hydrate, making it apparent that exudates are candidly involved in pollen hydration. Although lipids are fixed for having a role in pollen hydration, but there are evidences also which show that there are some other molecular moieties which have a role in pollen hydration. The mutation in the most abundant coat protein, that is the loss of oleosin domain JRP17 in Arabidopsis thaliana, delayed the pollen hydration ability by threefold as compared to the wild type. The delay in hydration is attributed to the inaccurate interaction of pollen with stigma owing to this mutation, which indicates that this protein may also have a role in pollen hydration. The pollen hydration also implicates stigmatic proteins called as aquaporins. Aquaporins are water channel proteins which, in addition to their role in cell incompatibility, also regulate the process of pollen hydration. Now, we come to the next stage of the interactions, that is pollen germination and pollen tube growth in stigma. After successful hydration, the pollen must germinate. The pollen tube emerges and starts growing in the stigma and then reaching the style. The initial penetration of the pollen tube into the stigmatic tissue at the outset varies from species to species. In Arabidopsis thaliana, the pollen tube has been found to penetrate the cuticle and then growing in between the cuticle and the main body of the cell wall till it reaches to the base of the cell. Then it enters the stigma transmitting tissue where it grows through the intercellular spaces. In members of family Asteraceae, the pollen tubes show extracellular growth to reach the base of the papillae, and then they progress through the middle lamellae of the cells. The lipids in the exudates are implicated for their role in directing the pollen tube growth by controlling the water flow to the pollen. The exudates are known to contain triglycerides as their major hydrophobic components. The hydrophobicity of the exudates is conceived to be a critical factor in the ability of the pollen to penetrate on the stigma. It establishes a gradient of water in the exudate, which is used as a directional or guidance cue by the germinating and growing pollen tube. After pollen germination of the stigma, the pollen tube grows in the style. Now, we will see how the pollen tube growth occurs in the style. After penetrating the stigma surface and growing through the intercellular spaces of the stigmatic tissue, the pollen tube reaches to the style of the pistil. In angiosperms, there are three categories of or types of style based on the morphological features of the style. These are solid style, semi-solid style and open or hollow style. The solid style is mostly found in dicots and is characterized by having a central strand of specialized and elongated cells which constitute the transmitting tissue, also called as the conducting tissue. The transmitting tissue varies in its complexity and may be either single stranded as in Withenia or multi-stranded as in Solanum melongena. The cells of the transmitting tissue are highly thick walled with intercellular spaces having secretions rich in carbohydrates, proteins, glycoproteins and some enzymes. The hollow style is predominantly found in monocots and consists of a hollow canal which runs throughout the length of the style from stigma to its base. A layer of glandular cells surround the stylar canal which secretes some mucilaginous substances into the canal which serve as nutrition for the growing pollen tube. These secretions contain proteins, carbohydrates, lipids, some enzymes such as esterases and acid or phosphatases, etc. After arriving in the style, the pollen tube grows towards the ori through the intercellular spaces of the transmitting tissue, where the elongated cells are piled into parallel files. This path of the pollen tube was earlier thought to be a mechanical one and determined by the transmitting tissue. But now it is known that there are biochemical cues which direct the pollen tube growth through the style. During its growth through the style, the pollen tubes are in addition with the components of the extracellular matrix of the transmitting tissue. The extracellular matrix, also called as ECM, consists of a complex mixture of proteins, particularly the arabinogalactan proteins, proline rich glycoproteins and extensin like proteins. The arabinogalactan proteins AGPs are highly glycosylated hydroxyproline rich glycoproteins. A class of AGPs called as transmitting tissue specific proteins have been known as the proteins which play multiple roles such as pollen tube adhesion, pollen tube guidance in the style and also the nutrition of the pollen tube. In experimental studies where the levels of TTS transmitting tissue specific proteins was reduced, the seed set was severely reduced, the severity correlating with the degree of dominant regulation of TTS protein synthesis. The reduced TTS protein levels resulted in the reduction of pollen tube growth through the transmitting tissue of the pistil. The TTS proteins show a gradient of glycosylation with their maximum glycosylation near the ovarian end of the style. Thus, it is proposed that the pollen tubes grow along this gradient of increased TTS glycosylation in the transmitting tissue. Further, the pollen tubes have been found to deglycosylate TTS proteins, releasing sugar molecules which are implicated for their role in pollen tube nutrition and pollen tube growth. There are other proteins of extracellular matrix which are implicated for pollen tube growth, such as NATTS, pistil specific extension like proteins, also called as PELPS. Precisely speaking, the glycoproteins in the style are believed to provide guidance to the growing pollen tubes. In hollow style of monocots, a cysteine rich adhesion SCA protein with a pectin fraction in the ecium are implicated for pollen tube adhesion, growth and guidance. Now, we come to the stage why the pollen tube has reached the ovule, and now we will see how the pollen tube will enter into the ovule. The pollen tubes, after growing through the style, reach the ovary and find their way into the ovules. The studies of pollen tube growth in many species have provided evidence in support of some chemotropic factors governing the pollen tube guidance. These studies have especially highlighted the role of female gametophyte in pollen tube guidance. The role of embryo sacs in the pollen tube guidance is clear from the studies on Arabidopsis thaliana, where pollen tubes were guided only to those ovules with normal embryo sacs and not to the ovules with degenerated embryo sacs. This clearly suggested that viable embryo sacs provide signals to guide the pollen tube. Studies in peach also have revealed that secretions from both ovary and ovules appear to have a role in pollen tube guidance and penetration of the ovule. In the ovary, the pollen tubes are guided to the ovules and may enter the ovule through micropyle, chalaza or integuments. The entry of pollen tube through micropyle is called as porogamy, while as entry through chalaza is called as chalazogamy, and entry through integuments is called as mesogamy. Now, we will discuss the entry of pollen tube into the embryo sac. Whatsoever is the mode of entry of the pollen tube into the ovule, the pollen tube always enters the embryo sac through the micropylar end. The pollen tube enters into the filiform apparatus through its apex and grows through it into the synergid. The synergid, which receives the pollen tube, appears to be predetermined as it starts degenerating well before the arrival of the pollen tube. After arriving in the synergid, the pollen tube ruptures and releases its contents, which include two male gametes or sperm cells. These two gametes are involved in the act of double fertilization. They are involved in the fertilization of the egg and secondary nucleus, forming zygote and primary endosperm nucleus respectively, which give rise to the embryo and endosperm in the developing seed or ovule. The pollen and pistil interactions we discussed so far are true of the compatible pollinations, which always result in fertilization and seed set. The angiosperm pistils have evolved mechanisms to discriminate between desirable pollen and undesirable pollen, which, if allowed to bring about fertilization would have severe negative effects on the fitness of the progeny. If a pistil with a functional female gamete or gametes fails to set seeds after pollination by viable and fertile pollen grains which can otherwise bring about fertilization in other pistil, the two are said to be sexually incompatible. Sexual incompatibility is of two types: one, interspecific incompatibility, and second, intraspecific incompatibility. In interspecific incompatibility, the pollen grains from other species are outrightly rejected because they are too dissimilar from the recipient species. The interspecific incompatibility is polygenic and is controlled by many genes. The second mechanism is intraspecific incompatibility, also called as self incompatibility. Here the pollen, which although originates from the donor plant of the same species, is rejected because it is too similar because of originating from a genetically same or closely related plant. The self incompatibility in most of the angiosperms is controlled by a single multiallelic S locus with genes encoding a pollen component and a styler component. The interaction of these S gene pollen and styler proteins constitutes the recognition and rejection reaction which determines whether pollen is compatible or incompatible. The self incompatibility is generally of two types: gametophytic incompatibility and sporophytic incompatibility. In gametophytic incompatibility, the incompatibility phenotype is determined by the genotype of the pollen or gametophyte itself, while as in sporophytic incompatibility, the incompatibility process is controlled or determined by genotype of the sporophytic plant from which the pollen is derived. Let us first talk about the gametophytic incompatibility. In species with gametophytic incompatibility system, both compatible and incompatible pollen are allowed to germinate and grow pollen tubes into the style. The growth of the incompatible pollen tubes is, however, arrested in the style once it reaches 1/3 length of the style. Analysis of styler proteins involved with the inhibition of the incompatible pollen tube revealed the occurrence of S-linked glycoproteins which have RNase activity. They are commonly referred to as sRNases. The sRNases enter into the pollen tube and their interaction with the pollen S protein determines whether to degrade the pollen RNA or not. In incompatible pollen tubes, the RNA is degraded, which brings about a cessation of the pollen tube in the style, while as in compatible pollen, the RNases are rendered inactive by the activity of the pollen S protein, thus allowing a normal pollen tube growth through the style to bring about fertilization. Now we come to the sporophytic incompatibility. In sporophytic incompatibility, the stigma is the site of rejection reaction. Following incompatible pollination, the pollen grains either fail to germinate on the stigma, or if germinate, the malformed pollen tubes are unable to penetrate the stigma, which develop callous depositions in the stigmatic papillae. The sporophytic incompatibility involves the sporophyte origin pollen coat proteins PCPs such as SCR, which interacts with the S stigmatic proteins such as SRK, S receptor kinase, and SLG, S locus glycoprotein. Following pollination, the pollen coat containing pollen coat proteins such as SCR flows to form a layer between the pollen and the stigma. If the SCR of pollen and SRK of the recipient stigma are encoded by the same S haplotype, the two are incompatible. In such an interaction between SCR and SRK, the inner domain of SRK phosphorylates the ARC1 within the stigmatic papilla, which initiates a cascade of intercellular signaling. This signaling cascade is suggested to ultimately regulate the activity of aquaporins in the stigmatic papillae to limit the availability of water to the incompatible pollen grain, inhibiting its germination and subsequent growth. To conclude, now we know that the pollen-pistil interaction is a complex interaction where a molecular crosstalk between the male gametophyte and female sporophytic tissue takes place at various levels in the pistil. This interaction governs whether the pollen may be allowed to bring about fertilization inside the ovules or may be inhibited from doing so depending upon the genetic nature of the pollen. Dear students, with this we come to the end of today's lecture. I take your leave and hope to see you in the next lecture. Till then, goodbye.